Well, calcium, like in the global south, it's iron and calcium, and I think there's another one, like, I think generally the overall protein content are the three things that are going down. So
Did you go
crops are
to
getting-
did you go to any discussion sessions with him? Yeah, today.
No, I saw it
Faculty
yesterday.
meetings with him?
No,
I'd love for wheat to be more of a C4 area.
yeah,
We're losing a lot, especially as
Fading.
temperatures go up.
Yeah.
We're-
Yields will go down.
yeah. I would like to, I mean, yeah, yields should go up if you can do it sustainably, but I would love to see the math on if the other narrative, like how, like if you reduced X% of corn and soybean that is not food for people, like if we stopped ethanol production in a totally like fiction, not fictional, but if you weren't feeding corn and soybeans to nearly as many animals and you didn't have ethanol production.
Well, that's a whole other issue that didn't get.
That's
yeah,
right.
That's a whole.
That's,
Yeah,
right, but that's the other narrative, right? Like, if we're gonna talk about feeding people, let's feed
No,
people
no,
and not feed
but what I would
plants.
say is,
Yeah.
that was why he was here. It would have been nice to hear his views on that, but he was here to talk to us about his science.
Yeah,
That's,
I think maybe that's why I didn't get into that.
yeah.
The closest I got into that was,
So what I'm saying, I did have those conversations
Yeah.
last night over dinner,
yeah,
and obviously in two. Consumption patterns are gonna have to be part of the solution,
Mhm,
even if
Yeah.
you increase the efficiency, you still gotta grow.
yeah. Super interesting. One of the conversations I had on my trip was about like food waste in nations like Egypt that are like, so much of their economy is based on it. And I'm like, I will stop and let us get to the topic at hand, but these economies that are based in tourism.
Yeah.
Because the resorts just have a huge amount of food waste.
Yeah, exactly.
That is a super interesting thing I've been chatting with people about the last week too.
Well, welcome, everyone. As you can probably hear, we're having a nice robust discussion here about how we can all get together and save the world. So thanks for joining us today. And as we initiate each of these updates and discussion meetings, we always open it up for any comments, input, ideas that anyone wants to bring to the table for the group. So I'll open it up now for any comments in the room or on Zoom. Anybody?
I don't have anything, but I would love to hear a little like synopsis of Egypt, Jess, if you want to tell us a bit about what you're doing over there and how it relates back to Forever Green.
Yeah, there's a lot to unpack there.
Well, just tell them where you were and why.
Yeah, so it, as you might know, the beginning of this story, that we were approached, I think a combination of us and the Land Institute were approached to do a blog post. Because the Land Institute is now part of the US cousin. I learned a lot more about these entities actually, just FYI, and I wanna, was the end of the story is that I like to maybe catch up with you, and I also have been in conversation catching up with Tammy about how all this went down.
Yeah, great.
So there's this whole, natural climate solutions entity that's global, and there's a US cousin, US Nature Climate Solutions, that approached us about doing a blog post toward COP27, which are, the COPs are these international climate discussions. And so, yeah, and then I decided to try to go, and then so I went to the talks as part of the UMN team that goes. So the Institute on the Environment kind of pushed for UMN to get observer status. So, these are like UN representations And they have this big climate talks every year. They actually have negotiations throughout the year. This is their big meeting of the parties. Observers are allowed, and so you can apply to get observer status, which is UNN, and there are lots of organizations and NGOs that have done this. And so, yeah, so I was part of the week 1 delegation. Another interesting thing I might mention about UMN's delegation is that it prioritizes graduate students. So of the, 7 week 1, 7 week 2, they divide the weeks so that twice the people can go. Jessica Hellman and I were the only 2 faculty, and so the rest were all graduate students, and that was amazing. Like 3 of the graduate students were Egyptian also, so that was
All right.
Wow.
so why I got to learn a lot about Egypt is that the Egyptian student with us in week 1 got us a meeting with one of the parliament members in Egypt to learn about what it looks like in that type of government to try to work on climate change and work on
I see, yeah.
sustainability. Super fascinating. So I got to be part of that. They're basically like, therefore 2 concurrent conferences. So there's like 3 tri-societies. So there's all the negotiations going on and trying to follow those. And then like Nature Climate Solutions and all these other entities have these different pavilions. The history of this is that the UN represented nations had pavilions, and that was their way of interfacing with observers in a little more public, like this is where John Kerry would come give a big speech, or where people would come give a big speech while all of their negotiators are in the negotiating groups. But it's kind of blown up into this big thing. I definitely have commentary on what I think of that thing, but the Nature Climate Solutions has their own pavilion also, and did programming the whole time. Most of the stuff I was supposed to be involved with fell through. It was not heavily organized. So yeah, so I'll reach back out to the comms people, but like did not have an interview. Like he didn't even organize the group of us. He made a group for us, and that was the extent of organizing. Like no one else's programming was on their calendar. Like it was kind of a pretty disorganized mess, unfortunately. I mean, there were struggles. Like they were literally still building the conference center when they were supposed to be putting their pavilion together. The internet was not good at all. He lost his luggage. So there were, I think he didn't get off to a good foot, but also it was not. Yeah, a lot of it fell through, but I did get to explore all the food systems stuff there and made some good contacts. And I attended a lot of programming at FAO's booth, and I was more impressed with as a UN entity, I was a lot more impressed with how they were bringing in different African heads of state. They're different people for much more rich conversation.
Cool.
Most of the presentations of those pavilions were just talking points, and they weren't that substantial. So
Yeah.
Well,
that's a super brief, and I'm still following. I'm happy to give I'm going to be giving my lab a more formal update, and I'm happy to do that for you all too. And I'm actually still following the results of the negotiations.
Yeah.
That's where.
Cool.
Okay.
Thank you
Yeah.
for the update on your trip.
Yeah, I guess I would say one more thing is that I believe they're thinking about having perennial grains be there more present next year. So that might be something to think about. Any of this perennial rice paper dropped the day after I got there, so I shared that. So anyways, so there's more to come in the future.
Okay, thank you. So today we're going to get an update from Brusa and David.
I'll just take over here, Don. You know
Very
me.
good.
Anyway, so I just wanted to say a few things. So Anthony's going to be giving a presentation today, but before he does so, I noticed Kevin's on the line and such, and I just wanted to kind of put in a blog to say that I and especially the program have been really blessed over the years by being able to attract really high quality genomics type individuals to the program. Kevin came in and really laid the groundwork for defining the gene space in Pennycress, and really got the whole program of genomics off the ground. So thank you, Kevin. After that, then Ratan came in and even third the program. And now Anthony is standing on the shoulders of those two folks, and we'll be giving a little presentation today, kind of a mix of genomics and trait discovery type stuff. And not to put any pressure on Anthony, I mean, I can say too, the folks that came before him, Kevin's now a USDA scientist at Fort Collins, and so he's done very well for himself. Ratan is now vice president of research at Pennycress. And so, Anthony, not putting any pressure on you, but you're up to bat, and I'll let you introduce yourself.
Thank you, David. Nothing like getting, no pressure right before the talk, right? But no, you do bring up a good point. Kevin and Ratan's work have been absolutely invaluable, and anything that is one of these long-running projects, you're going to be relying on the efforts of people before you, and you'll see exactly how important some of the stuff is that I'm building off of from them. So let me go ahead and grab PowerPoint.
Like I said, if you can introduce yourself a little bit too,
Yeah.
please feel free.
Yeah, so most of you probably know me from the work I was doing with Palmer Amaranth, but in 2011 I was doing sort of split time between finishing up Palmer Amaranth and starting to learn more about Pennycress and starting to get integrated into that team. And I was hired as researcher 5 in February of this year to work full-time, focusing on Pennycress trait discovery and development. Before we start, just make sure, is this actually advancing a slide for everybody?
No, go full screen,
That is,
presenter mode.
yep, all right. I've had that issue before. That's why I have a PDF version that always works. Okay. Okay, now do you see the slide?
No.
Was that a yes or a no? Sorry.
Yes.
Yes, okay, awesome. All right, so I'm breaking down the outline of this talk into sort of two main groups, looking at the actual sources of these new traits that we're going to be tracking down and identifying, and then the actual projects to figure out how to integrate them. So you have to find the sources of variation before you can even attempt to make use of it, basically. So sources of new traits sort of break down into there's three main groups that I'm working with, which are the actual wild populations. Very frequently, that means ones of Armenian descent, EMS mutagenesis, and the mutant gene index, which was recently released. In addition to this, other groups are also working with, for example, CRISPR constructs as another source of genetic variation. But these are the three that I'm specifically focusing on for the talk today. So within wild populations, I mean, this is basically where everything starts. You started with, wild pennycress that you found, and eventually we're trying to domesticate it and get it more and more economically viable. So if you look at the wild populations, you're going to find a lot of variation. And if you take the population genetics approach, the underlying principle behind that is when you get these isolated populations, they will eventually diverge genetically, and that's where you get variation from. And then when you have admixture and populations start crossing with each other, you can still track down the signal of that original source that the propagules came from. And when you're looking at these things, you have to keep in mind that you can have natural selection impacting things, you can have artificial selection impacting things, but you also end up having to deal with genetic drift. So all of these things can be influential in finding natural variation. So pennycress itself is native to Eurasia, but we find populations of pennycress as far away as North America, obviously, Chile, Tibet. I've seen we even have one from Greenland, apparently. So it's spread quite far. So to get a sense of what we were working with in terms of how much variation there was and which groups we should focus on for looking for divergence, I did a global population modeling of the populations used in the recent 2002 paper that was released. That's the Genome version 2 paper for anyone familiar with that. So the idea is to get a sense of where the variation is so that we can focus on tracking down valuable resources instead of wasting our time on things that don't have much variation in it. So when you go and you do the modeling, we did some k-means modeling, we did some structure modeling, which is Markov Monte Carlo chain. But basically it broke down into either 3 groups or 4 groups, depending on which model you used. When we compared BIC, we compared delta K values, they basically came out to be the same. So this is what it looks like if you break it down into a tree of 3 groups. And you can see in green at the very bottom that basal group is the Armenian descent line. And then group 1 and group 2 are basically monophyletic, very, very strong structuring with these 3 groups. And if we model it as 4 groups, our additional group, which you'll see in black, is actually distributed among groups 1 and 2 pretty evenly, which is sort of unusual to see that sort of structuring. But it makes sense when you take this data and you plot it on top of the geographic location, and it looks like this. So you can see in the middle there, you have in green, you have our Armenian group focused in Armenia, obviously, and group 1 in orange, mostly in Europe. And then group 2, we have a little bit more diversity of where it's located, but North America is the only location with group 3. And this basically visually depicts group 3 in black being a merger of the orange and the purple groups. So when we did principal components analysis and broke it down, that's exactly what we see. You can see here that the Armenian group variation is driven pretty much entirely by principal component 2, and that's an almost perfect 90-degree angle to principal component 1 variation that we see in groups 1, 2, and 3. So there's a very distinct separation for Armenian from all of the other groups, and this is all driven by basically just genetic distance. So Armenian is definitely high priority, but this also does illustrate what I was talking about with this black group here, group 3, being sort of a merger of group 1 and 2. And you can see all of the variance on principal component 1 is pretty much exactly linear, with group 3 falling right in the middle. So we get a good sense of what's happening in terms of the North American populations as they're shifting with these groups being introduced and bred with each other, and this Armenian focus. So takeaway from population modeling that I did, the Armenian populations are definitely have the largest genetic distance when compared against our reference, so min 106 ref. Those other remote samples, the ones located, for example, in Chile and Tibet, despite their distance, the fact that you would expect them to be isolated, there's not much genetic variation going on there. So they're either most likely more recent introductions, but there's not enough genetic variance to make those worth pursuing. And our North American cluster does appear to be emerging of 2 other groups into sort of a combined larger group. Second main source of variation is EMS mutagenesis. So this has been used for quite a while. So it's ethyl methane sulfonate. It's been used widely since the 70s. I believe it was first used in 1965 from what I remember. And it's very useful for producing a large amount of mutants very, very quickly in a single generation. And it's actually still widely used. And the American Chemical Society named it Molecule of the Week recently. So they still think that it's important. Very simple molecule down here, but don't get that on your DNA. Make sure you wear gloves and work in a hood. So the main benefit of this, in addition to really rapidly getting a lot of variation, is that we know the mutagenesis properties of EMS pretty well because of how much has been used. Specifically, most of the changes that we see are GC to AT transitions, making it much easier to pick out. And recent work has actually shown that the sites being flanked by GC also tends to increase a prevalence of mutations happening there. The real benefits for our work is the fact that because it focuses mostly on transitions, it creates point mutations that are very good for single locus traits. And then the third main source, this one is quite recent, is our mutant gene index, which is an absolutely massive collection of information on the EMS mutants based on M2s that were sequenced. And if you go through it, you're going to see that about 2/3 of it are going to be heterozygous calls, and 1/3 of it are going to be homozygous mutants. That's based on just sort of how F2 and M2s segregate. But that means that 1/3 of it are these homozygous mutants that are basically ready to work with. And 2/3 of it are hets that you'll need to do a little bit of work with, but are still definitely usable. And the most recent mutant gene index that I'm working with has 455,000 variants that are cataloged, and has a lot of additional very useful metadata, including homologous genes in Arabidopsis, so we have information on function, which is extremely helpful. The origin track, so that we can track down seeds and grow them out. And also information on whether or not the mutations are synonymous or non-synonymous, which can help with filtering as well. So that's sort of the stuff that we're working with. And then the second half of this is basically going to be focusing on individual projects that I've been working on to make use of those sources of variation. And these are the 4 main ones that I'm going to focus on, but there's been quite a few other things that we've got on back burners as well. So lead off with the big one, which is glucosinolates. Anyone working with Pennycress is well familiar with glucosinolates and is probably sick of hearing about them. But it's been one of our priorities is to knock down glucosinolate levels so that it can be used for human and animal consumption. They're very important plant defense compounds, but when they're ingested, particularly in the oils as we're interested in, it has anti-nutritional, goitrogenic, and off-tasting effects. It can cause a lot of problems, specifically with, in addition to gastrointestinal issues, with thyroid problems. So we have to be concerned about that. And it's particularly important with non-ruminant animals, and of course, humans are not ruminants. And in Pennycress, when we talk about glucosinolates, what we're really talking about overwhelmingly is synegrin, which you can see down there on the bottom right. So the main way that we've been able to address the problem of glucosinolates with the Pennycress team is by using a mutation in AOP2. And this is part of the biosynthesis pathway for aliphatic glucosinolates. And this mutation alone is able to reduce synegrin down to safe levels. However, whenever you're working with these types of things, particularly with a defense compound, there's always going to be these trade-offs. And the main problems that we see with AOP2, just the mutation on its own, is that it reduces seed size, and that has a direct impact on yield. The overall plant vigor has been reduced, and there's a delayed maturity issue. And since we're talking about using Pennycress in relay cropping systems, that termination date, the end of maturity, is very, very important for timing for getting soybean planted. Now, some work, and David can talk about this later at the end, but some investigation been done into what's actually causing the problems with AOP2, and it seems to be a plant-wide sulfur deficiency. And this knockdown of sulfur impacts the production of synegrin, because as you can see here, synegrin requires sulfur for its biosynthesis. However, it's affecting other parts of the pathway as well. So the obvious question at that point then is, what happens if you apply sulfur? And David has done some work on that, and we have some sulfur-rich fertilizer in the lab right now that we're doing some work with in the growth chambers, and it does cause recovery from these downsides. So it does seem to be the sulfur lack that's causing the problems. The main problems with just saying apply sulfur, though, is that's additional cost, additional labor, and the economics of that will have to be studied, of course, to determine whether or not it's worthwhile. But the other problem is this environmental impact, which is a big part of Forever Green. And there's this concern of, if you're applying this extra sulfur, is how much of that does end up in runoff. Particularly, this has been an issue out in California, where they use sulfur-rich fungicides on the wine vineyards out there, and they were having a lot of issues with sulfur runoff there. Now, we'd be applying it differently, but we don't know how much will run off, and it's something to be aware of. Particularly since a big focus of Forever Green is improving water quality. And sulfur specifically, if it does get into the water system, leads to production of hydrogen sulfide, which alters pH of wetlands, and also can lead to, through other pathways, increases in methylmercury, which is a neurotoxin that bioaccumulates. So it's definitely a concern. Sulfur pollution isn't something that's gotten as much focus on stuff like, for example, phosphates causing algal blooms, but recent paper showed that sulfur levels from agriculture have been the largest anthropogenic source. So it's something that we want to be aware of when determining what to do in this situation. So looking at all the stuff that's going on, My goal was to look for alternative traits that can reduce the glucosinolate levels without having these fitness and yield penalties and without requiring additions of sulfur. So we used a technique that's known as bulk segregant analysis, and it's based on the basic principles of F2 segregation, which is where the segregant part comes from. So just did a standard F2 creation using our low glucosinolate mutants that we found. These were EMS mutants crossed with MN106. Create your F1s and your F2s. And the idea is that the trait you're looking for should segregate as shown in the bottom. And we should find a homozygous mutant with our F2 low glucosinolate trait. So the question then, before we can even do bulk segregant analysis, is bulk segregant is focused on single locus segregation according to an F2 model. So does the phenotype actually fall out such that we would expect it to look like that? So in order to do that, we took our F2s that we were going to potentially sequence, and we used the NIR machine in order to estimate the levels of glucosinolates. So this is a density plot of the glucosinolate values after k-mean clustering with a means of 3. And basically this plots out into 3 distinct groups that fall out pretty clearly where you would expect our segregation to be. Our middle group, which would be our heterozygotes, is a little bit noisier, but this is still pretty solid. When we plot it out as a box and whisker plot alongside our control of MN106, you can see that it does fall into these 3 groups pretty distinctly. Most notably, the one that we're interested in down here, our low glucosinolate, shows roughly a 50 to 60% reduction compared to our controls over here, our wild type and our MN106 reference. So based on this, we're pretty confident that we're looking at something that's a single locus trait that we're trying to track down. So at that point, we just use growth chambers to grow out seeds from our F2s so that we can get higher yield DNA. And then we perform the bulking part of the bulk segregant analysis. So in this case, we had everything that we assume is our low glucosinolate. Well, our NIR told us was low glucosinolate. From each of those packets that you saw, we grabbed 10 individuals and then we did 10 packets each. So our final result for our low glucosinolates to be sequenced is this sample down here of pooled DNA from 100 individuals, normalized so that each individual is represented equally. We did that for our other 2 groups. And at the end of all of the sequencing, at the end of all of the extractions and all of the pooling and everything, we end up with 3 samples being sent for sequencing. But the 3 samples are representative of the segregation. So the underlying approach to anything like this when you're trying to do trait discovery is using models. And one of the strengths of bulk segregant analysis is it uses this F2 segregation model as sort of the null model that you're comparing against. It's a very simple model that you can make some very strong comparisons against. And the important thing that we're using in this case is allelic depth from our pooled samples. And allelic depth basically takes the equivalent of our sampling of just a regular statistical model. And there's 2 different tools that we investigated to make use of this. There's one known as MuteMap and another one that's an R package known as QTL Seeker. And both of them use delta SNP index, which I'll explain in a moment, but also QTL Seeker uses G prime, which is related to the standard LOD models. So I wanted to use QTL Seeker so we could compare against LOD for simple comparisons that should be more apparent to people in the program. But delta SNP also has a little bit more power for discriminating, so we wanted to make sure we could make use of that as well. So G prime is just a statistical distribution model that is directly related to LODs. In fact, you can just turn into LOD with a simple transformation command. So it enables us to set a threshold, for example, LOD of at least 4, which is a pretty common one, but it does tend to be a little bit less stringent. So we could look for G prime regions of interest as sort of a broader net. And if we wanted to be more stringent, then we look at delta SNP index. And this, instead of looking at likelihood values, this is looking at allelic depth ratios between trait and wild type pools. One of the unusual things with delta SNP index is it actually does have directionality. It can be positive or negative, meaning something can contribute towards or away from a trait, which is an interesting part of the modeling. And instead of getting the log odds values, we get confidence intervals when we use delta S&P. So to give you an idea of what the output looks like when we were scanning for this low glucosinolate trait, this is our G prime statistic with the bar at the bottom in pink that you can see that represents the same bar that would be an LOD of 4. So scaffold 1 seems to have something on it. Definitely our high priority. Scaffold 3 is extremely noisy and probably isn't going to be very useful. We've got a few other things that stand out as potentially being worth looking at, but nowhere near as strong as our spike at scaffold 1. However, when we look at it through the delta S&P index with our confidence intervals, we actually get quite a bit less going on. We still do get the same regions of interest in scaffold 1 specifically, and scaffold 3 is still a mess. But there's definitely fewer hits when we use delta S&P index. So the ones that we found in delta S&P index are going to be the ones that we're most interested in, particularly this positive one here in scaffold 1. Scaffold 6, the fact that this one's negative means it should be acting against the trait and interest, but it's definitely something that we keep in mind in terms of regions to investigate. So if we just looked at this region of interest, this is just what it looks like in IGV, and there is a lot of variation here. It's not too surprising. There's a lot of genes here, a lot of intergenic stuff as well. There's a giant pool of things, so we have to go through and filter this. And the thing that we're looking for, though, is something like this. And if we zoom in, this variation pattern is exactly what we would expect from a F2 segregation. Our low glucosinolate trait is a homozygous mutant. The intermediate value is heterozygous, and our wild type matches our reference. So this is an example of one of our high priority candidates that we would look for. So, so the region in chromosome 1 that we were interested in had 3,470 SNPs, along with some indels as well. And basically, to narrow it down from that, we did the stuff I just showed you. We looked at whether or not the traits were in genes versus being intergenic versus, promoter regions and stuff like that. And we had individual values for each of these variants for their S&P index values and their G prime values. So we went through and filtered through all of this. We reduced that pool of S&Ps down to 22 candidates in the specific region. So I went through and I started testing some of the markers, looked for the quality of the annealing sites and such, and we found a set of candidate markers that we ran, and this one that I'm showing you right here is the one that performed the best. But as you can see, very clear segregation across our 3 groups. But the problem is that this mutation and this marker is an intergenic target. It doesn't seem to directly cause anything of interest. So more than likely what's going on here is we found a marker that is in linkage with something that's actually causative, and we weren't able to identify the causative mutation yet. So the next steps for this reduced glucosinolate trait, we want to identify the gene that's actually responsible if we can. Having something in linkage is extremely useful, but actually finding the cause of it and being able to figure out what is causing it in terms of pathways is extremely helpful as well. So that's going to be the next thing we're focusing on. We also want to identify additional traits that can reduce glucosinolates. We have a few lines that are candidates for that. Keep in mind, the trait that I'm showing you right now reduced it by about 50 to 60%. That's not enough to knock it down to the levels that you see in canola, but it is still a significant reduction. If we can get another trait and stack it with this one, we could potentially get it down to that 00 level. So we're aiming overall for that sort of standard by combining multiple traits together. The other alternative is taking this 50 to 60% reduction and then using a processing step to remove some of it as well. But we're going to investigate to see whether or not we have any additional parts that are interested in. In terms of how close the linkage is, yeah, we do want to get a better sense of what's going on with the linkage. I'm going to be investigating things immediately in the near proximity of it, and also looking through the mutant gene index to see if we can find anything. But if we can find the actual trait, that would be priority. Working with the fact that it's in linkage is sort of the second fallback. Still useful, not as good. All right, so the next trait that I want to talk about is the stay green trait.
Anthony.
Oh yeah, go ahead.
Down here.
Has anyone ever determined what genes were responsible for the reduction in glucosinolates in canola?
Canola,
Right, it went from, very high levels, the Canadians took it down to 0. What genes did they, has anyone ever gone back and looked at the genes that are responsible for low glucosinolates in canola, going from rapeseed to canola?
As far as I know, I mean, there are candidate genes that seem to be suggestive, such as well-known genes such as transcription factors that are known to be involved in controlling the pathway genes. But still, I haven't seen that Nature or Science paper that comes out and says, this is the one. And so I would say it's still kind of up in the air. And let's apologize to Anthony here. I did not provide him with all the latest data. And we do have information now that the seed size issue with AOP, it's not AOP itself, it was something that was linked, and that we now have AOP mutants that have perfectly normal seed size. And I'm sorry I didn't get that to you, Anthony.
okay, so it was something in linkage though, we managed to break it, good.
Yeah, and the other thing that goes on too, there was a very informative paper that came out that was discussing the transition in canola from going from single zero, which is reduced uric acid, to double zero, combining the glucosinolate biz. And early on, they had extreme difficulty growing double zero canola that had reduced glucosinolates. And the fact that that glucosinolates contain sulfur, everybody kind of thought, well, these double zero plants won't need any extra sulfur because they're not making glucosinolates that have sulfur. But what they found in contrast was the plants have an absolute requirement for sulfur. And that is because, in addition to being a defensive compound, glucosinolates are also used by brassica plants as a sulfur reserve, probably in particularly in the seed. And so it's traditional, I mean, it's agronomically a normal practice to dump 30 pounds of sulfur onto the ground for growing canola. And again, I don't know, Anthony mentioned some runoff issues and things like that that are going to have to be dealt with. But I think we need to kind of take a look to see in the canola world better of how they deal with such issues as well. And that's something that we haven't had that discussion yet with the Pennycress agronomic team that needs to be done. With that, I'll let Anthony continue. Thank you, Anthony.
Yeah, no, thank you for the updates. It's definitely good to hear that there's been some progress on addressing it. There's still sort of this overall question of what is, going to be the agronomic priority when we're trying to get people to adopt it, and in keeping with, as I mentioned, the runoff issues need to be addressed and stuff like that. But it's definitely interesting to see just how many things are tied into these glucosinolate pathways. Tim, I just saw
Anthony,
that.
yeah. Can I actually also try to comment on Don's question with regard to what was known in canola, because that's obviously from early days of our effort to reduce glucosinolates we've been looking at, and in canola they did studies trying to map these genes that resulted in double zero. And the outcome of that has been published. So there are gene candidates. So some of them are more proven. So what we learned early on that there are multiple QTLs controlling that trait in canola. But canola genome is more complex as well because it's tetraploid and there are multiple alleles for each. But there are same genes as David has mentioned, some transcription factors that we early on were gene editing. And they do have an effect. They all have physiological issues or some other trade-off off-type effects. GTR2, which is not a transcription factor, but rather a transporter of glucosinolates, was involved. And there are essentially other minor QTLs as well with the gene candidates. Not a whole lot has been done to prove or demonstrate that those are indeed defining components of the 00 or low glucosinolate in canola phenotype. So there is limited knowledge in canola that can be translated. I expect in the end it's going to end up exactly like canola, multiple QTLs and breeding more for second site suppressors to reduce off-type effects. That's what my prediction is that's going to happen in Pennycress eventually. If it's okay, if Dawn asks a question, can I also ask a question? Or we should wait until the end? What do you guys
Sure,
think?
Tim, go, Tim.
Yeah. So I have to actually two questions. One is on the sulfur kind of rescue of the phenotypes. One also potential scenario is that you supply additional sulfur and the phenotypes go away or off types go away, at least some of them, but also your synegrin, glucosinolate, goes up. So kind of goes against your purpose. Have that been looked at? If you supply additional sulfur, is synegrin staying the same as the original AOP2 mutant?
That I haven't gotten as far yet, and so those studies are ongoing. But I can say for sure in the canola world that the application of extra sulfur still maintains the 00 standard.
Got you. So it is possible if there is some physiological role for glucosinolate per se, it's possible that by supplying additional sulfur, you're bringing up glucosinolate level, and that results in less pronounced phenotypes. But
I think more likely is that what happens is that the extra sulfur simply supplements and takes the place of synegrin or glucosinolates.
Yeah, that would be great. That would be awesome if that's demonstrated. Then my question is great progress on bulk segregant analysis. I would love to see the outcome of that. But one thing I wasn't clear, what was the other parent? One was MN106, the other one you called it low glucosinolate. What was actual mutant?
So that was from David's EMS work. We had a David, do you remember these specific? We had an internal tracking number from that, but that was from was on the X10 field, I know.
Yeah, I mean that particular mutant goes way back to 2015.
So David, my guess it's a mutant actual that we have on our joint patent application. My guess is 444P1, is that right? Or something like that.
I mean, you guys have options on 444 actually, according to our old exchanges. But in terms of the actual gene that 444 is actually mutated in, as Anthony said, and Rattan worked very hard to find it. He couldn't find it. He went through a bunch of different candidate genes, and none of them panned out. And so it seems to be something that might be novel. And so, of course, us being research scientists, that's the kind of thing we like as well.
So are you using this mutant trying to map the gene, or it's a different mutant?
444 is a mutant that we're beginning to bring into the breeding biz. And it takes it down. It doesn't go all the way down to a good 0. It goes down to 40, which is a little bit above where you need to be. It actually kind of takes it down to camelina range.
So what was it, 444 used in the bulk segregant analysis that Anthony just described, or some other mutant?
Oh, that was just a descendant of the original. And so we've been growing up that plant for a long time, doing crosses with it back to wild type. And so that maybe that's your question. Yeah, that was a backcross to the 106 population that he was looking at.
Well, I don't think I got the answer. Which other parent was used in a bulk segregation analysis in addition to MN106?
That was the original low glucosinolate mutant that we called for just field purposes E5444, which is just simply the plot that it was identified in.
I see. So E5444, which is in our patent application. Thank you.
Okay, I don't think it is.
Oh,
Because I mean, I think the patent application only contained mutants for which we had genes identified.
I thought it was mentioned, but I'll double check on it. Thank you.
Okay.
Okay, we had another sulfur question. How does sulfur fertility need change as you reduce glucosinolates since they're so sulfur rich? David, I was gonna say the sulfur stuff is fairly recent. We haven't done too much on it. So do you have anything to say about that?
There's quite a bit of published literature on it already out there on the canola side of things, and even on the Arabidopsis side of things. And indeed, if you take a wild type plant and you put extra sulfur on the field, you'll up the glucosinolates by 10% or so. If that answers the question.
Yeah.
I can say if you have a plant that's all as blocked in the pathway, sulfur isn't going to help that at all if you have a block in the pathway.
Anthony, let's turn it back to you.
Okay, yeah, we've got some ground to cover here. So for the second trait that I was investigating, we're looking at we call the stay green trait, and this is a picture from earlier this year. You can see the stay green individuals in the middle. Very clearly, you can see where the name of the trait comes from. So these are basically a delayed senescence of the leaves. So this is important because leaf senescence plays a role in source-sinks relationship between different plant tissues and transferring resources. And it's often in response to abiotic stress, drought obviously being one that we're particularly interested in, but also salt stresses and changes in light. And stay green has a history of being an important agronomic trait, specifically in grain crops. And so this is a plot from Duvik et al. This is looking at the use of stay green trait to increase grain yield in grain crops over time. It's been one of the major agronomic traits that was being bred for. And one of the main reasons for this is the fact that stay green traits have a longer carbon capture phase before they transition over into nitrogen remobilization. So potentially, depending on the stay green trait you're looking at, it can increase the yield of your plants, but not all stay green traits are the same. So there's, broadly speaking, there's two categories of stay green traits. There's ones that are called cosmetic and functional. And that depends on what the underlying mechanism is that's causing this longer green state. So for a cosmetic stay green, the mechanism is that there's something that's interfering or slowing down the chlorophyll catabolism and the breakdown that leads to the loss of green. So since the breakdown is delayed, it stays green longer. The remainder of the senescence pathway, though, generally remains fully functional in cosmetic stay green traits. Functional stay green traits, on the other hand, affect the entire senescence syndrome and actually changes the timing of the transition between major phases of the leaves. So this is a example of sort of the timing of the carbon capture and nitrogen remobilization phases. And senescence basically is swapping between the two main phases. And you can see you start getting that loss of green and senescence of the leaf. A functional stay green will delay this senescence to a later point in time. So functional stay greens are complicated because they're involved in a lot of different pathways, most notably major plant hormones that are involved in it, including ethylene, abscisic acid, and jasmonic and salicylic acids are related to it, and they play important roles, as I mentioned, in stress response. And specifically, we are interested in response to water shortage. So relating stay green to drought tolerance. So because senescence allows for remobilization of resources, it is a response to these drought pressures. Stay green traits will still senesce under severe drought, but that response is delayed. So if you get a short-term shortage, the leaves will not throw the switch that causes them to go in senescence and eventually destroy the leaf. So you can get a better tolerance for short-term drought through functional stay greens, not cosmetics. In addition to that, beyond the resilience, you also have stay green potential for use in higher biomass production because of that increased carbon capture phase, which would enable it to be used, for example, high energy forage crops. Or there's also the potential out there for using pennycress as a green manure. So you plow it into the ground to give you some improvements to the soil health, and the high glucosinolate levels in this case could potentially be a benefit for control of soil pathogens and pests. And this is already being done with mustard green manures. Washington State University has been working on mustard green manures for about 20 years at this point, and pennycress actually matches pretty well with the guidelines that they give. So when they're, you can see here, this is one of their releases, and they're talking about the main traits that they're looking for green manures. You're looking for high biomass production and a low seed cost, obviously from the economic perspective, but high glucosinolate concentrations are actually a benefit in this case. And you also want lack of weedy characteristics such as dormant seed, which we have actually addressed in pennycress through the TT2 and TT8 mutations. So just looking at the characteristics of what's out there, there's definitely a potential for using pennycress as a green manure. So for figuring out what is actually happening with our stay green traits, we did bulk segregant analysis, very similar to what I just showed you before. We sequenced some F2 stay greens and some wild types that were part of that F2, and we used QTL seeker again. So for this one, this is the exact same thing. This is the cutoff of LOD equals 4, and you can see we've got quite a few hits in this case. But when we ran it through delta S&P index, however, we got absolutely no hits. And this is because the two approaches use very different underlying statistical models. So we didn't really have anything to look at in terms of delta S&P index. We did investigate, however, the regions that were flagged by looking at the LOD scores. And we had a few candidates, but so far none of the candidates have performed particularly well in terms of our genotyping tests. And my suspicion on this is that it was most likely due to phenotyping error of the original samples that were sequenced. And one of the main issues we have is that our stay green trait, we showed you the picture from the field and it looks very distinct, but when we were doing it in the growth chambers, we had some difficulties with that. Kevin, go ahead.
Yeah, so I think QTL-seq plots a transformed delta SNP index. Have you looked at the untransformed delta SNP index to see if that lines up with your p-values?
Do you remember what the transformation was?
I don't remember. It's been a couple of months since I looked at it, but I did notice the same thing in a sugar beet study that we've been doing. So yeah, that transformation masked some relevant stuff. Okay,
I definitely I was looking through it and I didn't see that in the manual, but I went back and double check. I may have missed it, but the thing to note here is that our delta S&P index, even if the transform would rescale things, the majority of hits we're getting in this delta S&P index are actually negative values, so working in the opposite direction of what we would expect for the trait. So that's why we use two different approaches, and we just basically went with the LOD scores in order to investigate our targets. But I will definitely go back and check that and see whether or not transforms give me some more useful information here. So yeah, I'm definitely thinking that because we had some issues with the stay green phenotype not being as apparent and easy to quantify when we were growing them in the growth chambers, that we may just have not had the best selection for our bulk segregant pools. But because of the potential for this and the strength of the field phenotype, I think that it's still worth pursuing. So we're going to try, I plan to do bulk segregant analysis again with better phenotyping to sort of, improve the quality of it. But also, I'm going to be investigating the mutant gene index for stay green traits as well, and looking through what lines we have for that. Which brings us to part 3, which is looking through the mutant gene index and tracking traits of interest out of that. So as I mentioned before, mutant gene index is 455,000 mutants, and they're all derived from EMS M2s. We have a lot of useful information on gene homology that we can use to track things down, and we have a lot of filtering tools that we can make use of. And the main strength of the mutant gene index is that we have this really, really large useful resource, and it becomes even more powerful when we combine it with pathways that are well characterized in the literature. So for this is an example of a leaf senescence pathway taken from Schipper's 2015 paper. And you don't need to look at the specifics of this, but the idea here is that there's a bunch of these different pathways relating to different plant hormones, and each of the genes that are listed here, mutations in them could potentially affect leaf senescence. And if we take this figure, take other things that already have been discovered in terms of pathways, and we combine it with the mutant gene index, that enables us to narrow things down. And I was able to identify down to 900 mutants that have some type of mutation in genes associated with these pathways. There's definitely a lot more filtering to go. 900 is quite a bit, but that is sort of the initial first round of filtering that I did to look for additional stay green traits. One thing to keep in mind, though, when looking through the mutant gene index is that when you're working with M2s, not everything that you get in in your seed packets is going to be what you expect. We were using EMS, and EMS causes mutations in individual cells in the M1. And those cells have basically different lines of descent within the plant. So potentially different branches of the same M1 plant can end up having different mutations present in it. And this was something that Ratan looked into. Thank you for looking into this. The math behind this one was very involved, I'm sure. This was looking at separate individual plants. So each of these sections represents a different branch of the same plant. And this is just a Venn diagram showing the variations shared in common. So from this individual plant, we only had about 3,000 variants that were shared across all of the branches. Whereas, for example, this branch up here had 9,500 variants that only it had. So whenever you work with anything from our mutant gene index, you have to be aware that you're going to be looking at situations like this, because all of the seed in these packets, the seed that we're using and storing, comes from all of the branches, even though only one of them was sequenced. So what does that actually mean for us when we're trying to use these M2s from the mutant gene index? Well, any given seed might not actually have the mutation that we're expecting, but there is a pretty simple way to address this. We know the target of the mutation. We know, thanks to genome version 2.0, exactly what the region is surrounding it. And we can easily design markers to genotype to see whether or not an individual has the trait in question. So essentially just it's a numbers game. We need to grow enough seeds and then genotype them that we have enough plants to work with going downstream. So we're estimating somewhere roughly in the ballpark of about 50 plants. After genotyping, we should still have a decent number to work with. And then the last, hang on, let me catch up with chat real quick. Okay, we'll return to those later. We're at close to the end here. But there's definitely some interesting stuff in chat to discuss. So Armenian Pennycress populations, we already know, I showed you before, they're very genetically distinct, but we actually had an even more genetically distinct one. Generally, the Armenian populations are divergent, but they're still close enough related that we can cross them with 106 or with one of the other lines. And David identified an unusual line that was Armenian that does not cross with 106 and has some distinct physical traits that are very different. Most notably, it has fewer seeds, but the seeds themselves are larger. The overall yield is comparable. Basically, it's a break even for that. They flower earlier, but the maturity date is still the same. So it takes basically longer to go through the process. Larger, fewer seeds. There is reduced chatter without us introducing a trait for it. And the flowers are larger and noticeably more fragrant. And of course, there's this inability for it to actually cross with 106. So I did some phylogenetic work on this just to sort of give a sense of what we were working with. And you can see here, I basically broke it down to the point of origin, all of our Armenian ones in the red box, and then our MN breeding groups down here in green. And our reference right here is clearly labeled. So what happens here is that we do actually see our Armenian group is what we call polyphyletic in this case. It is not all just one Armenian cluster. This Armenian pooled sample, which is our one that we're investigating, and ARM116 are very distinct compared to the other Armenian samples and to all of our MN lines. So whatever's going on with these two is very, very strong genetic isolation driving genetic distance. So it's definitely something unusual that we notice, and we definitely want to pursue it because there's definitely a lot of potential here. The first step would be to see whether or not we can do reproductive isolation breaking. So if we cross this the sample that we have with one of these other AIM samples from Armenia that is closer related, can we still get them to actually produce seed? We also want to try and identify high impact genes that are driving the divergence. What is actually causing this incompatibility between them? Whether or not there's specific genes responsible for it that we can identify. And there is sort of this underlying question of what is the taxonomic status when you have this separate group that's not able to actually interbreed? Is it a separate subspecies? You know, how do we classify this? And what are the legal implications of that in terms of intellectual property? So with that, I'll do a quick round of acknowledgements, and then we can get caught up on chat with all of the interesting questions that people have been fielding. So big thank to the entire UMN Pennycress team. I'm just a small part of this. I'm doing trait discovery. Traits have to be, introduced into lines, and you have to move forward. And the traits that we're interested in also get informed by the rest of the team. So there's a lot of back and forth, and it's really important for this work. So thank you to everyone on the Pennycress team for all of your efforts. And specifically, I want to thank Samantha Wells and David Marks for basically onboarding me in 2021 and getting me my introduction to Pennycress, which I was completely unfamiliar with outside of, forever green presentations before 2021. I also want to thank David specifically for all the mutagenesis and the M2 and F2 creation. Those things were basically in the works before I was hired in February, and I was able to just basically springboard off of that and immediately start launching into some of the genomics work on it. And of course, the Anderson and Marks lab, basically for all of your advising, particularly for a lot of the MSI stuff and streamlining those pipelines, and basically just for being my research home for this year. And obviously for Evergreen, iPrep, and I prefer all of the extended collaborators on that are extremely important, and thank you all for your input. And special mention to the team that worked on Genome version 2.0. This was the paper that came out earlier this year. Basically everything in Pennycress discovery moving forward is going to be using these resources. So I just needed to give a shout out to everybody on this gigantic list for all of their contributions to that, and also for giving me a chance to make my contribution to that paper as well with the population genetics work. And with that I will take questions, starting with chat. Hang on, let me pull this up real quick. Mitch was brainstorming with Carl Rosen about Pennycress as a biofumigant green manure for potato.
Yeah, what you were saying, Anthony, just made me remember that conversation. I was talking to Carl Rosen, and they use other mustards, spring mustards that have been bred for high glucosinolates in those systems, since they have so many disease issues, and it just seems like a perfect fit to fill that role, that plant protection role, with a winter cover crop instead of spring. And obviously the winter cover, there might even be a benefit of being able to get potatoes in the ground sooner. I don't know if that's a good thing in that system, but it's worth looking at. And then thanks, Kevin, for the suggestion that could also be relevant to sugar beets. I didn't know it. Is that used widely in sugar beet systems, Kevin?
No, not at all, but I think with the prevalence of fungal pathogens being the primary yield drags on sugar beet, that is, I mean, any biofumigant could be a potential game changer, because we're running out of chemical control options there.
Yeah,
And just to go
I mean-
back, and David has been telling us that he has some Pennycress lines that have very high levels of glucosinolates. Is that right, David?
I mean, certainly there's potential. I mean, I think that even what Anthony just showed, that looks like he might have identified a negative regulator of anthocyanins, or excuse me, glucosinolates. That might be something to pursue. That one that when he was looking at the bulk segregant analysis and it dipped down, that might be an interesting gene to pursue as something that might increase glucosinolates. And overall, I mean, my thoughts on this and why I was thinking green manure is exactly what you guys are talking about in the potato world, in the sugar beet world. That is to be able to use a winter cover crop to fill that bill.
It seems though you hear me? It seems though that's indirect. Like, you'd have to make a choice though, right? Like, if you had a high glucosinolate line, it might not be fit for human consumption.
Yep, this would be
Well, to back up, when you do a green manure, there's no harvesting of the seed at all. What you do is you simply take the green plant and you plow it into the ground. Yeah.
Exactly. Yeah. Exactly.
So we just have to have a couple of different lines in the breeding program. Yeah.
I would be curious though. I mean, it sounds like this is done, but yeah, I'd be curious to know like the pounds of sulfur that you get from these high glucosinolate lines compared to some of the like fumigants, the sulfur based like fungicides that were used. I mean, it's also an interesting thing, like, because the lack of sulfur fungicide is like one of the hypotheses of why in the last decade or so we have more and more sulfur deficiencies in crops, because we're not throwing all this extra stuff out there anymore, and we don't have like, we cleaned up coal and other things that
coal power plants.
Yeah, coal is a big one, and just in general, the fact that you're harvesting something and removing the nutrients every single year.
So there's a demand for a new pollutant source.
Well, the interesting story about the whole 0.0 is that the paper that I actually connected with that filled me in on what was going on was that early on when they made 0.0 canola, there was no issue at all. And this was in the, at a time when they were just changing over the coal plants to reduce the high sulfur burning coal. And so the environment was packed with sulfur already, and they had no issue. And then as they improved the coal plants, that's when they saw their issues.
Exactly, because who needs forests?
Okay, bring back coal.
Acid rain, yeah.
Bring back coal.
It is funny that that happened, because that was one of the things when I was investigating this that kept coming up when you're talking about levels of sulfur in the environment. Is they kept calling back to the issues with like acid rain caused by sulfur, and that one paper I mentioned saying that agronomic sulfur sources have surpassed fossil fuel at this point, and part of it is due to the fossil fuel output of sulfur being reduced by being cleaner. But part of it is, the agronomic use, particularly when we're dealing with some of these challenges like fungal pathogens.
Jim Anderson had a statement here.
Well, I mean, at least, applying sulfur containing fertilizers is much more targeted than acid rain going everywhere. So I guess David or Jess, do you know how much sulfur they're putting on a canola crop? And at those rates, I guess, Jess, if you have an opinion, is that, something?
My understanding and read is 30 pounds in the spring, as the plants are beginning to bolt.
Maybe more.
Or the other way they do it is that they'll put in, so the plants use sulfate. And so the other way that they'll do is they'll put elementary sulfur onto the soil in the fall and let the bacteria convert that into sulfate. But like I say, the spring application, and I can't tell you, we're using ammonium sulfate. And I think that's the typical, and somebody might know better than me of what they're adding on. But and so in that case, you get kind of a boost of both the nitrogen and the sulfate.
Well,
I guess, Jim, I would wonder, I was just gonna say, I would wonder, I mean, because sulfur, I mean, sulfate might, but is sulfur gonna move like nitrogen does?
Yeah.
Like, I mean, sulfate might, because it's also negatively charged, but that would be my question, is like, if you have to apply it, like, is it moving out of the system? Because the idea of a green manure to give you the sulfur, but yeah.
Sulfur deficiency has been a little bit of a concern in wheat for more than a decade now, because the glutenins have amino acids that are high in sulfur, cysteine and methionine, I think are the two. So if there is a sulfur shortage, the bakers see that, like what happened to the wheat that you guys used to grow that was very strong gluten, it's getting weaker. And we don't think it's the genetics, it's the environment.
Is Nancy Elke on Zoom today? She leads the Canola Research Center in Minnesota. Nancy, are you on?
I don't think I saw her. I have time too.
Well, Nancy would be the person, one of the individuals that would know the current production system, and she could provide some input in terms of the use of sulfur in canola production here in Minnesota.
Yeah, you're right.
Les, you've got your hand up. I know that you've worked on water quality issues a lot. Are you wanting to chime in on this point?
Well, what I was going to say is that sulfur is being added in the rotation anyway in many places now. And so putting it on ahead of cover crop would just be changing the timing of the application of the sulfur. It wouldn't, as far as I see, it wouldn't be a big problem. I think they're putting on like 20 pounds for, you can check that, but I think it's around 20 pounds for corn in places where it's being put on. I think if you want to check on sulfur dynamics, you might want to check in with Paul Bloom in terms of off-site problems.
Oh, yeah, that's right. Yeah.
Yeah, you're right, Jim. So you may have problem.
Any other comments or questions for anyone online today or from anyone online today?
We did have one from Nicholas Jordan in the chat we didn't get to, which is the stay green trait viewed as flatly problematic in relay cropping with pennycress, or might the stress tolerance features provide a benefit? So that is one of those things where if you're extending it, we do have to have that termination date. So that was one of the things where we're trying to sort of look at that in terms of if we can do like earlier maturity, earlier flowering would be sort of a combination just to shift everything instead of having it run long. David, did you have any thoughts on that?
I mean, really, like I say, the stay green biz is really just kind of a separate play than the oilseed play, in my mind. If we find out something interesting about any kind of stress tolerance or any kind of biological information that will allow us to make a better pennycress, then that's a bonus for me.
That's true, because if if this is used for example, for the green manure, or if it's being used, you know, as forage, then you're not necessarily looking for the complete maturity and drying down of the mature seeds.
Yeah.
You're basically
And
looking for biomass.
Yeah, and I would say too, in such a situation that it will mash better, even mash better with the following traditional crop if we just plow the stuff in whenever we need to.
Yeah, so if we wanted to use stay green for oil seed production, then we would want to pair it with something to shift the maturity earlier. But if we're looking at it for any of these other potential applications, then the stay green trait on its own would be beneficial without something else. That's sort of the takeaway.
Stay green could also be helpful in an oilseed context in areas of the world that are not as limited seasonally. So if you're not trying to cram it into a tiny window like we have to do in Minnesota, you know, stay green might give you the yield enhancing benefits that you get in other crops. Obviously, we're focused on Minnesota, but the potential of those traits is there for other applications.
Absolutely, and Don always poo-poos this idea, but in any case, you know, out west and other places, camelina is just grown as a straight up spring planted summer crop, and we have spring varieties of pennycress that this could be bred into that could easily fit into those windows as well.
Yeah, it's interesting to look at all of these options, and as we all know, you know, we have a special focus here in Minnesota, but this technology, you know, as we are suspecting, is going to have application all over the world over the next decade. So stay green, you know, doesn't necessarily fit, you know, the oilseed part, but it certainly does in the state, in the green manure and other environments in the US and around the world. So
Yeah, and you mentioned, you know, possibilities. Trait discovery is all about possibilities, but this discussion we're having right now, and you know, hopefully a bunch of future discussions, will be really useful for sort of getting a sense of what those priorities are, because there's a lot of different ways this could go, as we've just discussed. But very useful to get this input from everybody. So thank you for everyone who's been asking questions.
The only feedback that I would give you is also discover the yield gene.
I mean,
and the gene for growing up through like 6 inches of corn residue.
And
That
the
would be
genes
helpful.
will allow it to go through residue, right?
I mean, I could just skip the middleman and look for the profitability gene.
There you go.
Well, Anthony and David, thanks for your great update and presentation and all the excitement that you have going on in your team. Really appreciate your presentation and the time you took to present to the group. So thank you. And remember, next week is Thanksgiving, so we won't see you on a Friday until in December. So have a happy Thanksgiving, stay well, and we'll see you in December. Bye-bye.
Yep. Thanks,
All right,
Anthony.
buddy.
Safe travels.
Thank you.